Hi, everyone, and welcome to another Trial Site News interview. I'm your host, Brandon, and today we'll be talking again to Dr. David Weissman and Daniel O'Connor. Dr. Weissman is an experimental pathologist with a background in pharmacy, pharmacology, and immunology. At Johnson & Johnson, as one of the only 66 research fellows, he led a research program where his duties included pharmacovigilance. His work has focused on fibrosis and pain, running his R&D company since 1996. His COVID work includes dataset analyses that have overturned previously negative studies used to justify public policy regarding hydroxychloroquine and ivermectin. He has made over 36 submissions to government bodies, including the FDA, CDC, and NIH, on aspects of COVID-19 pro-vaccine safety, efficacy, manufacturing, DNA contamination, and regulation. Daniel O'Connor is the founder of Trial Site News, and you can read his articles and opinion pieces there. Recently, he appeared on Chris Cuomo's TV show News Nation earlier this week and has been involved in the debate regarding safety data of the COVID-19 vaccines. Dr. Weissman and Daniel, thank you so much for joining us today.
Thank you. Good morning.
Thank you.
Today's discussion will cover the FDA analysis of safety data regarding the COVID-19 vaccines and the information gathered from a FOIA request by ICANN, Aaron Series Group. Dr. Weissman, do you want to start us off here?
Well, thank you for having me, and I'm honored to be interviewed by an emerging celebrity, Daniel O'Connor, fresh from his appearance in the Chris Cuomo program. And everything that I'm going to be talking about is capsulated in this title. So if you do nothing else but read the title, you can go home. Safety signal loss and underestimation of potential risks and suspected adverse reactions due to truancy, masking, and filtering in FDA's analysis of VAERS data associated with the COVID-19 pro-vaccines. And just so you know, this will be the subject of a paper which is going to be, by the time this show airs, will hopefully be published or posted as a preprint on a preprint server, and no doubt the trial site people will provide a link to it. There's four foundational things that you need to know to understand what I'm going to be talking about, and these are terms that have been sort of bandied around. They need a little bit of explanation so that everyone's on the same page. So we're going to be talking about a statistical signal. And a statistical signal is declared when the number of reports in VAERS for a particular adverse event, let's say myocarditis, occurring after a particular drug or vaccine, let's say Pfizer, meets certain statistical criteria. It doesn't matter what they are, but they have to meet certain criteria, and then we declare a signal. But a signal means that we can reasonably suspect that drug X may have caused adverse event Y, and the term suspected adverse reaction is a term in the regulations applied to investigational new drugs. So it is a real term, suspected adverse reaction. But that doesn't prove that the drug did it. We need further investigation to establish that drug X actually caused adverse event Y. And it's important to note that statistical signals are actually safety signals. FDA might try and say otherwise, but they're wrong. And these represent potential risk. That's a legal word. The FDA is required legally to consider for an EUA, emergency use authorization, without needing to establish causality. So that's an important point here about what we're going to be looking at, what they are, and what their importance is, and what their sort of legal standing is, as it were. And as you mentioned at the beginning, the ICANN organization put in a FOIA request, probably I think 2 or 3 years ago, for one of the types of analyses that the FDA and CDC do to analyze whether or not there's a signal. And this type of analysis is called an empirical Bayesian data mining analysis. And when the FOIA disclosure finally came out in January, consisting of 153 pages in a PDF, this in the middle part here is a typical listing of all the adverse events for which a signal had been detected. And I've just sort of expanded the bottom part there just to see, so that you see that each line represents a different type of adverse event. And I've colored in Janssen pink, Moderna yellow, and Pfizer green. And you can see that the pink, the Janssen 110 signals outnumber by many fold the eight yellow Moderna.
Dr. Weissman, can I just chime in for a second?
Yeah.
Yeah, I see where you're going, and I'd like to just step back and provide a little context for a minute. So just
Okay.
bear with me.
Yeah.
Now, this FOIA, Freedom of Information Act request came from Informed Consent Action Network, ICANN, which is Del Bigtree and Aaron Siri, their attorney.
Right. Right.
Correct? That's correct,
Yes.
right?
Yes.
Now,
yes.
A question. I want some context for the audience here. What we had access to VAERS, so what additional information from VAERS did ICANN need a FOIA for, first of all?
Okay, that's a good question. So, okay, so VAERS is a publicly there's a
Yes, we know that. Let's just talk about what was not available that they needed to file a FOIA for, submit a FOIA for.
Okay, so the FDA and CDC have to take the data from VAERS and then do some maths on it in order to determine whether or not there is a signal
Right.
for any type of adverse event, and they do different calculations to determine whether or not there's a signal. One of the types of calculations is called empirical Bayesian data mining, which requires you can't just do it on an Excel spreadsheet. You can do it for the other type, but not this type. You need software and computation to do it. And so FDA were charged with doing this analysis and then putting out reports, I think pretty much on a weekly basis, if I'm not mistaken. And the FOIA request was to request those reports that FDA was supposed to be generating every week. So
okay, I have a question. I get it. I have a question. Why wouldn't, that's public information, why wouldn't they just provide that?
Well, when you say it's public, I mean, it was internal to FDA. Yes, they should be providing it, but they didn't. So why wouldn't they? Exactly.
Okay, so we established that there was public information. We pay for this as taxpayers.
Right.
It's our health. Their job is to protect our health. Correct?
Right.
And ICANN requested this information. The agency didn't give it to them.
And they put up a fight. I mean,
they put up a fight. So
yeah.
When you've put up a fight for something, that means that there's something going on there, obviously. So you get this data. Now, does this data represent all of the VAERS entries? Because remember, very few individuals got the Janssen shot. Most people got Pfizer or Moderna. Later on, maybe a little Novavax, but by far, overwhelming number of vaccinated in the United States was either Pfizer or Moderna. And most of the injuries in VAERS, and again, VAERS is not causal, doesn't mean that the vaccine caused it.
Right.
But most of the reports in VAERS linked to Pfizer or Moderna. And what you're saying here is this interim report where agency employees that took this data, crunched the numbers in a statistical package.
Yeah.
The output was this massive Janssen signal.
Right.
And that at a high level, we're just establishing that. That's weird.
It is. I mean, so you've jumped in there, and I mean, you don't need PhD in statistics to see that the pink is 9 to 22 times more than the yellow and the green there. It just jumps out at you, and that's before we correct for the point that you've just made, that the Pfizer was used about 8 or 9 times more than the Janssen, and Moderna was used about 5 times more than Janssen, roughly on a per person basis.
But how can you? Sorry, I'm just going to get into this. I mean, how can you explain this? I mean, how can there be? I mean, it's the product was barely used.
well, it wasn't barely used. I mean, there were still 14 million or something people, or 50, in that sort of number, as opposed to dozens or hundreds of millions. But it wasn't like a few people.
Well, there's 230 million full under the category fully vaxxed, meaning
Right.
You received your primary series plus a booster. There's 230 or 240 million people.
Right. So it was about for 15, from my recollection, when it sort of plateaued out of Janssen, and let's say it was about 90 to 100 million of Moderna, and then the remainder was Pfizer. So do the math. That comes roughly to the 280 that you've just mentioned. But so you sort of asked about three different questions there. So I think that we can, if I've missed some, then you'll get me back. But the main question I think you're asking, which was the nub of what we're trying to discuss here, is that this imbalance just doesn't look right. It just does not look right. Okay, and anyone can see that, especially given that this is before you adjust for the relative usage. When you adjust for the relative usage, this 9 to 22-fold times that I'm showing at the top there becomes closer to 100 times more signals for Janssen over Moderna or Pfizer. That if FDA really believed that these numbers were correct, then they should have done something about Janssen way before they did. It should have been taken off the market very soon. These sorts of numbers.
It depends on the signal. I mean, if the signal is, "Hey, you got a headache," and it went away in an hour. I mean, it depends on the severity.
It does, but when you're seeing 100 times more, and again, to get into the weeds, when some of the signals aren't just headache that went away for a few minutes, or an hour. I mean, that some of the signals, and you can't appreciate it from this slide, were hematological in nature. Clotting types of disorders and so on. Way more than the thrombosis, thrombocytopenia, which was the subject of which triggered this pause in Janssen.
Yes, that's right.
In like April 2021, and so when that was based on 6 cases in the Janssen, no cases in the other 2, which was certainly an imbalance. But it was a glaring difference. But there were other kinds of events that were much more numerous, and the signals were pretty strong. For other kinds of clotting disorders, and FDA sort of didn't talk, and CDC didn't talk about them. And again, my analysis, talks about all those signals and what else they knew and when they knew it. But even if you just stop here and you just look at this picture, and you say something is wrong with this analysis, this simply does not make any sense, right?
Well, it does. But let me just jump in here. I want to make this lively and dynamic and interactive.
Yeah.
It does make sense if it's the truth, and if the reality is that Janssen is just that much more of a problematic vaccine.
Well, okay, so that's a very good hypothesis. So the rest of my study examines that question: was Janssen that bad? Was it really that bad?
Yes.
And so. Do you want the spoiler alert, or you want to wait until the end here? What do you want?
Well, I think that given the amount of information flowing around the internet, it's good to, I think, go ahead and talk about what you found, and then let's get into some of the details of the study.
Okay, so. I mean, you're raising really good questions. Was the question, really, to sort of rephrase it perhaps, is, was Janssen really this bad? And was Pfizer and Moderna really much better? And that's really the nux of the crux of what I was trying to figure out. So if I'm just going to show you the 124 signals that I just showed you for the 3 vaccines here in FDA's analysis, and I'm going to explain the terms as much or as little as you want later on, but I'm just going to show you the bottom line to sort of get us going. So if we basically figured out that signals were missing from that analysis. So Pfizer and Moderna were missing signals, and the calculation, which I can get into later on is that we estimate there are about 281 what I call truant signals. They just were not there. I don't know why they weren't there, but they weren't there. Okay.
How do you just sorry to jump in? How do you know that they weren't? How do you know that those truant signals from Moderna and Pfizer were there?
Okay, well, that's getting into the weeds of how I did the calculation. So how I did the calculation was I compared the FDA's Bayesian analysis with other analyses that were done by other with credible groups, and I
Okay.
compared the relative proportions of the different vaccines in those analyses, and then I corrected using those that information and come up with the number 281. That's the sort of top line of what I did. Okay.
Got it. So you, just to make it understandable, you took sort of comparable analyses
Yes.
and with the same products,
Yes.
and you found different results.
Correct. And then so when I sort of adjusted them, as it were, or made them comparable, to make them comparable, the only way I could make them comparable was by coming up with 281 signals that just disappeared. Okay. Don't know why. Okay. So that's step one. That's step one. So even if we stop here, right, that just sort of gets us back to the previous question. And the spoiler alert here is that if you notice the relative sizes of the red Janssen, yellow Moderna, green Pfizer,
Right.
in the previous one, you can see the proportions are very different.
Right.
Okay. So actually the size of the red is the same. It's just that the yellow got bigger and the green got bigger, because that represents the truancy of signals that were there. So so if we stop there and say, okay, well, Janssen, it was still fairly bad, but not as bad comparatively as we thought, in the previous slide. And when you do the maths, and it's not shown on this slide, when you would do all the adjustments and so on, that Janssen actually had, instead of a 9 to 22-fold excess of signals over the other two, they only had a 5 to 9 excess, which is still
high,
which is still not good, but it's not 22. So I think legitimately we can say that Janssen was producing more signals, okay, than the other two, which on its own should have evoked much earlier action than was actually carried out. But it wasn't as bad as the others, so it's still not good.
Well, it depends on when the data was collected. So if a lot of this data accumulated later on.
No, it was starting to show up, pretty early. Yeah.
Okay.
All right. So if we stop here, then this is a pretty bad situation, just to lose 281 signals out of a total of 405. But it gets worse, because the way they did the analyses was problematic as well. And the first problem they have, again, I can go into much more detail, but on the top line basis, there's something called masking. And masking means that the signals for one vaccine interfere mathematically with the signals with the other vaccine. Okay, doesn't matter how that happens, but they interfere. One interferes with the other. It's a known problem. Yeah. So when you adjust for this problem, you find another 365 signals. So now we're up to 770 as opposed to the 124 that we had before. Okay? Right. And then there's another problem, and the other problem is that there's something called filtering. And filtering is a different problem from masking. Filtering means you just set the threshold in an inappropriate way. And so when you adjust for that issue, okay, we get another 117. And now we're up to 763 missing signals. And it doesn't end there, because there are borderline signals which should also be mined. Remember that this technique is called empirical Bayesian data mining. Okay, that's right. The word that's used. So right. When you're mining, you should mine sort of equally, as it were. So there are borderline signals that actually may turn out on further examination to be real signals. Okay, and there was in this calculation 580 borderline signals, some of which don't know. It could have been 5, it could have been 2, it could have been 10. I don't know, but there were some number of borderline signals that probably would have come out to be real signals. And of course, some of the signals that I've shown you in the red, yellow, green may actually have been false positives as well. So, but you have to do an equally zealous investigation of the positives to see if there are any false positives, and the negatives to see if there are any false negatives. And that wasn't done. Okay, so the sort of the bottom line to everything I'm going to say, and then we can get into as many weeds as you want, is that there were hundreds, and actually, other calculations that I've done get it up into the thousands of safety signals were lost from FDA's Bayesian data mining analysis through these three mechanisms: truancy, masking, and filtering. These represent mostly uninvestigated potential risk. FDA was required legally to investigate and to consider for the EUA without needing to determine causality. Potential risk was an important element of the equation that they were supposed to have followed when they authorized the EUAs. And so, again, we can get into some detail. This is a really important point, that the different standards were applied, different evidentiary standards were applied to the assessment of safety and efficacy. That's really important. And then lastly, of course, if what I'm saying turns out to be true, these anomalies impugn the reliability of regulatory decisions and vaccine compensation decisions. And as well, of course, VAERS has been touted by CDC, FDA, and so on as the nation's early warning system for vaccine safety.
Right.
And it genuinely could be, but it clearly failed. And you're on the board of React 19, which is gratefully appreciated, I know. And recently they published or posted different interviews or meetings that were held over a period of time with Peter Marks, who was the head of the vaccine, or the Bureau Center for Biologics. And in one of those meetings, or several of the meetings, he's saying,
right,
we didn't see any signals for this, we didn't see any signals for that. But a very telling remark comes from one of his staff people, Dr. Naya, who was discussing, whether they could find signals or not. And he says something like, my instructions from Dr. Marks are to leave no stone unturned in trying to find, these signals for what the React 19 people were talking about. Well, okay, that's great. But clearly what I'm showing you here is that, that's great if you say that, but clearly that wasn't happening. Okay. And that's the nub of it. So everything you need to know is here. Now I can get into as many details.
Well, let's again, this is all very complicated, and there's so much information circulating around. At the end of the day, they took the raw data, they run this statistical analyses, and they come up with a extraordinarily high Janssen safety signal.
Yeah.
They didn't do anything with it. They
Right.
didn't go public that we know. I mean, I don't know of anything that they did.
Correct.
They didn't want to share this. There had to be a legal fight to get the data. What are the real world implications? What is how does that help people, patients?
Well, I mean, it doesn't. I mean, we as you've written about, nuance and in some of the articles you've written recently, and your interview that you did with Chris Cuomo a few days ago, the public needs to have trust in the public health institutions, FDA,
right.
CDC, and so on. And on the one hand, if they're being told, oh yeah, this is safe and effective, and this is the most studied vaccine, and so on and so forth, right? And then we find this is going on. I mean, if that red, yellow, green bar that you asked me about at the beginning was true, something should have been done about it. And on a graph that I've got later on, if you want to see it, is that I plotted those ratios over a period of time, and they remain fairly stable. And the points on the graph, in fact, I'm just going to try and find it real quickly so that I'll show it to you. This is one of the graphs that's showing you. This is the blue here is a Janssen, and
it's staggering. It's just staggering.
It's persistent. And then if you look at it here, this is every dot on this line. So we've got the blue is the Pfizer, and the red is the Moderna here. This is the ratio of how many more Janssen there are compared to the other two. And I had a number between 9 and 22, so that comes from this graph. Each one of these dots represents a point in time where there was an FDA or a VRBPAC or a CDC ACIP meeting. And at those meetings, almost, I think every time pretty much, there was a report, usually from CDC, usually from Tom Shimabukuro, but could have been other people as well.
You attended a lot of those, and we've
interviewed about those. So
a few of them, yes,
many of them. So the safety report was being given regularly, and not once, to my recollection, was any imbalance of this type being discussed. I mean, wouldn't you think that would be a pretty important topic to discuss, as opposed to just saying, oh, well, we had 6 cases of thrombosis and thrombocytopenia?
Well, let's look into that. Is there any reason why the FDA would not report this?
Well, I mean, the reason that comes to my mind, again, it's speculation on my part because I haven't seen any emails to that effect and so on, is that if this was known, let's say, let's take a step back. Eventually, Janssen was taken off the market. It was voluntarily withdrawn, or at Janssen's request in early 2023, I think it was. Okay. If Janssen would have been taken off the market in April 2021, it would still left the other 2 vaccines, but it would have had a huge impact on vaccine hesitancy. Because look at the converse. The converse was that, if you recall, in August 2021, the Pfizer product was given a full approval, a BLA approval, as opposed to the EUA authorization. Okay, now that approval actually didn't make any practical difference because that product, the Comirnaty, was never available, at least not until much, much later. But it was never available. The only product you could get at that time was the EUA version. They were legally distinct products. That was the terminology that was the FDA used in their documentation. But here's the thing: at the ACIP meeting that followed that approval or authorization, the CDC said in a slide, and I think we might have talked about it in one of our earlier interviews, is that they had done market research on vaccine hesitancy, which they felt was a huge problem. And one of the sort of conclusions they came from that market research was that they found that that people were hesitant because no product had been fully approved. Okay, but if you approve one product, we don't care which one it is. We will take any of them. So if you approve Pfizer, okay, I'll take Moderna, I'll take Janssen, I'll take Pfizer. Doesn't matter which one it was. So by approving one product, it supposedly reversed vaccine hesitancy. So if that was true in the sort of positive sense, right, how much more so would it be true in the negative sense? In other words, if they took off a vaccine, that would cause vaccine hesitancy to skyrocket.
I have a couple of questions on this. One is, David, did pharmaceutical manufacturers have any influence over how adverse event data was classified?
By classified, you mean like
grouped or reported? I mean, was there any influence of the drug companies on any of this data?
I don't know. I'm not aware of specific examples of where, let's say, Pfizer, for example, said to the FDA, "You need to be analyzing the data this and this way."
Okay.
The only incidents I can think of that remotely comes close to what you're describing is when the Janssen product was paused in April 2021. There were 2 ACIP meetings like a week apart. Mm-hmm. And one or both of those meetings, of course, the Janssen company was invited to give a presentation as well. And basically they sort of said something like, from memory, they sort of supported the decision, even though there's no causality has been proven, and this is an abundance of caution and those sorts of words. So in that sense, they were making their case publicly. What they were saying privately, that would be a great FOIA. We'd love to see those emails and records of those conversations, but I don't know that we have anything like that. Maybe
is it, assuming your study is correct, and assuming that your comparables are correct, okay?
Right.
What explains this? I mean, either A, Janssen's far more dangerous. B, was there any political, economic, or interagency pressures to downplay or acknowledge or defer certain risks?
Well, I mean, we know in general, there was efforts. Anytime anyone brought up any safety issues in public, whoever they were, that they were jumped on. In fact, there was an instance in the documents that were released that Senator Johnson released, from his subpoena in his capacity as the chairman of the Permanent Subcommittee on Investigations. There were a number of documents he released in June. And in those documents are minutes and proceedings of one of the vaccine safety technical group meetings. That's a sort of subgroup of ACIP. And in some of those meetings, some FDA people had done analysis of CMS data, and they were finding signals that were for, I think it was pulmonary embolism and some other things, that they could see even, let's say, February of 2021, and the minutes of the meeting sort of reflect that everyone was jumping on the FDA guys. Oh, you've got to investigate this more. You've got to, this is not, ready for publication. I can't remember the exact words, but the tone of what you read from the minutes is that within the agencies, even FDA people, when they were finding signals,
right,
everyone was jumping all over them to say, well, we've got to be really sure of this. And there's another email from Peter Marks, again in that same disclosure,
right,
in May, I think it was, of 2021, where he's questioning, that finally CDC was acknowledging myocarditis, even though signals, as from my analysis, that we can get into the weeds, but that those signals really did exist as early as February. The Israelis were telling the FDA and CDC, we're seeing myocarditis signals, and they were sitting on it and procrastinating on it and so on. So there was clearly efforts to suppress that, and it was really suppression because if you remember, May of 2021, the Pfizer teenage, like 12 to 18, I think, or 12 to 17 age group was approved or authorized for EUA use. Okay, that information to get out and to be acknowledged as a signal was critical for the deliberations of ACIP at that point, and that was clearly being suppressed. Okay.
On that note, in this study, and I get that, and I agree, we've covered that. Did the FDA use methods to aggregate or smooth out this data in ways that dilute rare but serious outcomes, perhaps with Pfizer and Moderna?
Well, sort of. So aside from the issue of just truancy, where I can't explain why there are no signals there, but in the mathematics, the mathematical methodology, yes, there were two procedures that were deficient in the general idea of what you've just asked me, and the two things are masking and filtering. So just, do you want to explain masking in a bit more detail?
Yeah, why don't you just high level for the audience?
Highlight it. So
masking versus filtering.
Okay, so we're talking about masking. So masking here, high level, where signals for a vaccine of interest are hidden by the presence of other reported vaccines. And so basically what was happening, the events, let's say myocarditis as an example, for Pfizer's myocarditis events got added into Moderna's control group, and Moderna's myocarditis events got added into Pfizer's control group. Okay. But is that sufficient? Do you understand that now, or do you want me to go into more
well, I, again, my understanding is, masking. It's the data is technically there, but it's somehow statistically obfuscated, or
because of this. And when you bump up the control group artificially, you're now reducing the difference between your target vaccine, your vaccine of interest, Pfizer, and you're making the control group look much worse than it really is, because you've added Moderna into the control group, but you haven't told anyone you've done that.
Yeah. See, but so filtering is the data never makes it into the analysis.
No,
in the first
that's
place.
A different. Well, hang on, we haven't got to filtering yet.
No, right. No, I get it. You explained masking, and
okay, so that's masking, and okay, so you're good with that. I've got other explanations, but if you're good with that, we'll move on. The point about this, about masking, is this is a well-known problem.
Right.
And part of my analysis
right.
is that in the other type of method, which is called a PRR method, which also the CDC put out, and we've analyzed those data as well, although they're deficient in other ways. But in general, when you calculate these PRR methods, PRR numbers, you also you need to account for masking. So what I've done, I've gone in and I've done the adjustment for masking in the PRR. But in the Bayesian, you need computation to do that. The thing about the Bayesian is that there was a paper which I'm going to show you in a moment from the. Let me show it to you. I'm going to show you the three sources because you asked me about comparators, and this is probably a good time to talk about that. Okay, so in this slide here. The top is the data set that we're looking at, this Bayesian FDA as well as the CDC's safety similar type of analysis using this other method called PRR, and the comparator populations or data sets that from which we've drawn all these conclusions, been able to draw these conclusions, are what they call the Oracle data set, which was based on a paper by Harpads in 2022. Whose authors, and this is really, really important, the authors of this paper are Dr. Dumichel, who was the inventor of this empirical Bayesian data mining method. Okay,
interesting.
And also on that paper is Dr. Scharfman, Scarfman, who is in the FDA. She's a leading biostatistician. And 25 years ago, she championed and worked with Dr. Michel to bring in this empirical Bayesian data mining into the FDA for them to use as one of the standard methodologies to look at drug safety in general. Okay, so this paper is really, really important. And what's really even more important about this paper is that there's a table they provide in their appendix of these different numbers and these signals for 7 adverse events. And here's the thing, the focus of the paper is masking. And they point to a version of this Bayesian data mining, which again, Dr. Dumichel and colleagues developed, which generates a different kind of number, but related number that takes account of masking. And they show the differences between masking and not masking using those methods. That methodology is available in the software the FDA was using, right? And FDA had the software to account for masking, and they didn't account for masking.
Right.
Okay, they could have done it. They had the software to do it, and they had Dr. Scharfman somewhere else in the FDA, not in the vaccine, section, right, to advise them on how to do it. It's not. I mean, I'm sure it's not that difficult to people who know how to use this type of software. But they had the resources to do this, and they didn't do it. Right. And so the Oracle people actually showed, and I showed 700 whatever missing signals, but they show a calculation, based on the entire VAERS as of October 2021, US and foreign reports of 24,000 masked, 24,000 potentially masked signals in VAERS. 24,000. Now, I couldn't get it that high. I could only get to about 7,000. I couldn't get to 24,000. But these are staggering numbers. And this is Dr. Dumichel originated the Bayesian mining method together with other people within the FDA acting in their non-official capacity. There was a disclaimer in the paper to that effect. So that was one source. And the other source was this data set, which is called the Violin, which I call the Violin data set, which is an online tool that anyone can go to. There's a website there, and it calculates these PRR values. It's a similar type of value to the Bayesian data mining. And so we extracted from that database all the signals that were existent as of that particular day, April 2022. And then we were able to look at the effect of masking, look at the effect of the threshold issue.
So Dr. Wiseman, given we're getting close to the end, a couple comments, questions. One, the bottom line seems here that that the masking we've observed, that your study observed with the FOIA data,
right?
One, it could represent the result of a combination of technical design considerations, statistical competition bias, institutional caution. Is that possible?
I mean, all of those things. I mean,
a reluctance to elevate early signals.
Yeah, I mean, I just want to. Can I just show? I get see one other slide. Okay, there were two things: masking and filtering. All right, and this slide addresses filtering. And filtering means you set the threshold, like you're moving the goalposts. Basically, that's what that means. And so, this slide here in the middle is taken from a presentation from the FDA to this vast committee, which is the Vaccine Safety Group, which is sort of a part of ACIP, I think, given on April 2021. And they're explaining to that group how they do this Bayesian data mining. And they say here, technically, any EBGM value, which is the number, the magic number they generate, above 1 indicates disproportional reporting, means that above 1, that becomes a signal. But we're going to use the quote unquote standard threshold of 2. Okay. So they're admitting here that really we should be using 1, but we're actually going to use 2. And I've given you 2 sources here. One from the same Dr. Schaffman that was on that Harpaz paper, who's writing, I think in 2004, saying, well, even though people are using this threshold of 2, which predates COVID, it's by no means optimal. And these other people are also FDA people. This value of 2 is arbitrarily chosen, depends on the context. And if you look at the pharmacovigilance literature, again, this predates COVID. This is not specific to FDA, right? In this particular story, is that the pharmacovigilance people are always concerned, well, if we generate too many signals, we'll never be able to do any work. So we have to balance. We have to balance that we can't generate too many signals. But in the context of, well, we've got a brand new drug that's never been tested before under an EUA, we have to be not leaving any stone unturned, as Dr. Nair said. And again, in this Harpaz paper that I've quoted, they use a threshold of 1. So Dumichel and Schaffman in this 2022 paper are using a threshold of 1. Okay. And so if you're representing that we've got enhanced surveillance and VAERS is the early warning system, et cetera, that's just simply not true.
Right. Not the case. Yes. And with that, I think, because again, I have to actually leave. So in conclusion, from a scientific, you've put together a wonderful paper. We're going to make this available. We're going to write an article about it. We will share it with friends that may have influence here and there. What, from your standpoint, what are the next steps that investigation can be done so that we can figure out what this very large anomaly is?
Well, I mean, we got to have access to all the records and the deliberations that what went on. You've asked questions. What were they saying? What was their reasoning? And we've tried to get in the minds of the people without having all the information. So that has to come out.
Okay.
We need to be talking to, well, Dr. Sharfman. Like, well, wasn't anyone talking to Dr. Sharfman? I mean, she sounds like she's a pretty reasonable person and could have given very valuable insight. And that would have been an important question. We've certainly got probable cause. I mean, the data, of course, by its nature, can't be perfect, but just that initial graph that you stopped me on at the very beginning. I mean, that's alone there is just probable cause to say we really need to be investigating this.
Well, I want to say this. This is a very important.
Yeah.
The stakes are big. President Trump has raised the topic of the vaccines and safety.
Right.
He has declared that the pharmaceutical companies have told him everything's great. The efficacy and the safety are perfect or near perfect.
Right.
We know that's probably not true. We know that there's quite a few injuries. You found something here, and there is an investigation now. So what we want to do is raise awareness about this study that you have done, share the methodology, and point out that there definitely could be, we're not sure, but there could be some maneuvering mathematically to again filter or somehow
filter, mask, or remove. I mean,
filter, mask. That's right, filter, mask, or remove.
Yeah,
this needs more attention. So if I think we should there be interest in further pursuing this, let's say sponsors are interested, where can people find you so that we can continue this?
I've got to thank you very much. Well, they can always find me through you because
of course, yes,
which I appreciate.
But we want to let everybody know.
But I've got my Twitter handle at adhesions.org. They can look me up on Twitter and message me there. And if someone's interested, they will find me.
Yes. So I think,
and of course, when the paper comes out, there'll be an email address on there which you'll post.
That's right. We'll do a report on the paper. We'll break it down and describe in more lay reading terms,
right,
what the study is, what you did, and what are the issues.
Right.
This is good scientific investigative work, Dr. Wiseman. It's important that we start to look at what was going on. We know ACIP is investigating this right now. We know President Trump wants an investigation into these products. And any last minute comments before we go back to Brendan?
Well, I just want to give you sort of a sound bite that, people, because unfortunately we have to condense everything into words of, in Twitter size things. And so to summarize, so people like to remember that the following: hundreds of COVID vaccine safety signals were lost in FDA's Bayesian analysis. You can add due to truancy, masking, filtering. And these represent potential uninvestigated risks that FDA was legally required to consider without needing to determine causality. Okay,
right.
That's really important because everyone says, "Well, you didn't prove causality." We don't have to prove causality. FDA didn't need to prove causality, but they needed to at least publish what these signals were, and they weren't doing it. We've got other examples which we've talked about in other interviews in the past, and the paper goes into some of those examples as well.
Right.
That these signals were being, they need to disclose that there are signals, even though they may turn out not to be real signals, but they need to be disclosed, and they weren't.
Yes. So this is, I think, incredible work. Again, Dr. Wiseman, you've done as much as anybody I know investigating some of the, what we think could be subpar scientific work associated with the government, the vaccine program. But we don't want to make claims. We have to keep investigating this situation, this question. Thank you, Dr. Wiseman. Brendan?
Thank you.
Yeah. Dr. David Wiseman and Daniel O'Connor, great discussion today. Really appreciate it.
Thank you very much.
Thank you.
And for unbiased news and opinion articles on the latest in scientific research, please visit trialsitenews.com. Also follow us on YouTube, Rumble, and X.